Hello everyone. In this particular video, we are going to explain about the secondary structures of protein, different secondary structures of protein, how the secondary structure is formed from the primary structure of protein. So let's start. So this is the four level of protein structure, and the first level is the primary structure, which is nothing but the linear sequence of amino acids. These are the single letter abbreviation of defined amino acids like T means tyrosine, A means alanine, G means glycine, P means proline, or D means glutamic acid. So when the proteins or is made up, the protein is made up of different types of amino acids, and when the protein is in linear form, that is there is no such type of interaction occurring between the different amino acid, then it is from the primary structure. That is, the primary structure is nothing but the linear sequence of amino acids. When this defined amino acid ultimately forms some type of interaction between between the amino group, amino and carboxyl group, between one amino acid with that of the another amino acid, that is located far ahead from the primary sequence. Say this T is interacting with the Y sequence, this S is interacting with this R sequence. So this is, there must be some sort of intermolecular or intermolecular hydrogen bonding. Then it forms the alpha helix or beta plated sheet, or this is known as the secondary structure of defined amino acid. So secondary structures of amino acid forms when the linear sequence of amino acid ultimately forms some types of interaction between intermolecular or intramolecular hydrogen bonding. So after the polypeptide chain or protein has been organized into successive stages of the secondary structure, such as alpha helix or beta plated sheet, apart from that, reverse turn or other loops can also be present. Combination of such structure elements, this is the alpha helix and this is the beta plated sheet. So this is the combination of such structural elements that arrange first into distinctive groups called the supersecondary motifs. And when these supersecondary motifs ultimately forms a domain, and the domain actually arranged on the same polypeptide chain in three-dimensional configuration, it is from the tertiary structure. So tertiary structure is the spatial arrangement between the domain on the same peptide chain results in an order 3D form of the protein, and it is the final native form of the monomeric protein having single polypeptide chain. That means if the polypeptide chain is composed of more than one subunit, it is ultimately from the quaternary structure. So quaternary structures is the unit of the tertiary structure aggregate to homo or hetero multimers. Not all protein have the tertiary structure, such as hemoglobin. Hemoglobin contains four different polypeptide chains, such as alpha 1, alpha 2, beta 1 and beta 2. So they can form the quaternary form, but those protein only that have only one one polypeptide chain or single polypeptide chain or monomeric protein, they ultimately form the tertiary structure. So in this video we are going to explain only the different types of secondary structures that is found in protein. So there are three types of protein secondary structure that can be found in protein. These are the alpha helix, beta plated sheet, and the random coil. So this is the polypeptide chain. This is the polypeptide chain. When the polypeptide chain fold into a zigzag motion, that is this particular structure or that structure. So this is the zigzag structure. It is called the beta plated sheet. But when it forms a coiled like structure, this is the coiled like structure, it is called the alpha helix. And the random coil is located between the alpha helix or beta plated sheet, or between the one alpha helix with that of another alpha helix. Random coil is actually made up of those amino acids, they cannot form truly alpha helix or beta plated sheet conformation, such as a stretch of aspartic acid, a stretch of lysine, or a stretch of bulky amino acid, which cannot form a favorable interaction to form ultimately this alpha helix or beta plated sheet. So they can form the random coil structure. So random coil is the third type of protein secondary structure. Next, this is the alpha helix. So we can see in this picture that alpha helix is a rod like structure. The tightly coiled polypeptide main chain forms the inner part of the rod. The atoms are in van der Waals contacts across the helix, thereby maximizing their association energies. We can see these the R groups of the each amino acids are located outward from the helical backbone to avoid static interface of the polypeptide backbone when with each other, because if the R group is located internally, in that case there must be some sort of clashes between the amino acid, or positive charge or negative charge. That is why the R groups are always located outward from the helical backbone. A single turn of the helix, this is the turn, this is the one turn of the helix, start from that point to that point, this is the single turn. This single turn of the helix extends about 0.54 nanometer, which is also known as the pitch of the helix, and it contains approximately 3.6 amino acid residues per turn of the helix. Alpha helix is stabilized by the hydrogen bond between the CO of nth residue with the NH group of N+4 residue. What does it mean? It means, say, this is the CO group. Sorry, this is the CO group of one amino acid. This will make a hydrogen bond with the NH group of another amino acid that is located 4 residue ahead in the linear sequence. So if we count this, this is the one amino acid, that is R2. This is the second amino acid, this is on the third amino acid, this is on the fourth amino acid. This is the fourth amino acid which contains the NH group. So this NH group is hydrogen bonded to the CO group of amino acid that is located 4 residue ahead in the linear sequence. Similarly, this CO group is hydrogen bonded to another NH group. This CO group is hydrogen bonded to another NH group. That means all the mentioned NH and CO groups are hydrogen bonded to each other. Thereby alpha helix is pretty much rigid. You cannot rotate the alpha helix. That is why it is written that is the helix is rigid. So the hydrogen bonding prevents free rotation. Here the hydrogen bonding, all the main chain NH and CO groups are hydrogen bonding, and this hydrogen bonding prevents free rotation, and that is why the helix is rigid. The NO distance, this NO optimum distance is about 2.8 angstrom. An alpha helix can form either with D or L amino acid, but the residue must be one or only of either L or D stereoisomer. Naturally occurring alpha helix can be made up of either right-handed or left-handed alpha helix. So if it is left-handed, so this is the left-handed, we can see there. This is the left-handed alpha helix. The left-handed means it is the anti-clockwise rotation. We can see this is the anti-clockwise rotation, and if it is right-handed, this is the clockwise rotation. So both left-handed and right-handed alpha helix can be found in protein, but in most of the cases the right-handed alpha helix are predominantly found in protein. Left-handed alpha helix are generally not found. So the distance between the two turns of the helix is known as the pitch. The pitch is about 0.54 nanometer. You have to remember this value. Now we have to explain about the stability of alpha helix, how the alpha helix gets stabilized, or in when case the alpha helix gets broken apart. Imagine a situation that the alpha helix, there are several amino acids located quite close enough, and that is actually made up of only similar type of charged molecules such as glutamic acid. Say glutamic acid, aspartic acid, glutamic acid, aspartic acid, aspartic acid, glutamic acid. These are present in close proximities. That means all acidic amino acid present in close proximity, or basic amino acid are present in close proximity, like lysine, arginine, histidine type of amino acids are present in close proximities. In that case, they will not form an alpha helix at P7 because of the strong repulsion between the negatively charged group or the positive charge group, which is sufficient to overcome the stabilizing effect of hydrogen bond on alpha helix. So that is why it is unstable if the polypeptide chain has many adjacent similar charge amino acid. That means if it is made up of glutamic acid, they will not be able to form a strong hydrogen bond alpha helix, or if it is made up of lysine, then it is not be able to form the alpha helix. Simultaneously, it is unstable if a polypeptide chain has a long chain R group. That means the bulk and shape of certain R groups can also destabilize the alpha helix or prevents its formation, such as aspartic acid, serine, threonine, leucine. These are the amino acids whose R group is bulky. They tend to prevent the formation of alpha helix if they occur close together. And the third point, that is the proline is the helix blocker. So you can see this is the proline. Proline is the not true amino acid, rather it is called amino acid because it actually contains NH group instead of NH2. So say this is the normal peptide bond. So when normal peptide bond occurs, what you have to understand here, in normal peptide bond, normally one amino acid contains NH2 group, but in peptide bond formation, this one H atom will be eliminated by the formation of water. So only one NH group is present. So that NH group can get hydrogen bonded with the CO group. This is the point. This is the, if NH group is present, they can form hydrogen bond with the CO group, so that they can form hydrogen bond, so that they can form actually the alpha helical structure. That means the two amino acid can come close together. But if proline already contain one H group, during formation of the peptide bond, proline do not have the free NH segment. So the proline do not have the H group, so they are not able to form any kind of hydrogen bond with the available CO group present before the fourth amino acid ahead of the linear sequence. In that case, they are not able to form the alpha helix. So this is the reason that why proline is the helix broker. And number two, because nitrogen is the part of the rigid ring, so constraints of the constraints on the formation of alpha helix is the presence of the proline residue. So because the proline nitrogen is part of the rigid ring, and rotation about the NC alpha bond, this is the NC alpha, this is not possible because the this is the NC alpha, because if you rotate this bond, the whole proline molecule will gets rotated. So rotation about the NC alpha bond is not possible. That is why proline is the helix broker. So this is the stability of alpha helix. Now we should move for the another aspects of alpha helix, the different types of helix that can be found in protein. So apart from alpha helix, 310 helix or 2.27 ribbon or pi helix can also be found. These are the different types of helix. And in order to understand what is the alpha helix or what is the 310 helix, what do you mean by 310 helix, we can write a formula that is n by m, where n is the number of residue per turn of the helix and m is the total number of atoms including hydrogen. That means if we consider alpha helix approximately contains 3.6 amino acid residue per turn of the helix, and we can write alpha helix as 3.6 by 13, because it contains 13 atoms including hydrogen in its complete turn. Similarly, if it is 310 helix, that means number of amino acid present is 3 per turn. We can see there are 3 amino acids, red colored, 3 amino acid residues per turn of the helix. And it is the 10 means the total number of atoms present in 3 amino acid including hydrogen. If it is the pi helix, in that case the number of residue per turn is 4.4, and this is the structure. This is the 4.4 amino acid residues. So we can understand how the 310 helix can form, or how the pi helix can form. And alpha helix can be rewrite as 3.6 by 13, because it contains 3.6 amino acid residue per turn of the helix, and it is a 13 amino acids. Simultaneously, if there is a question that what do you mean in 310 helix, that means 310 helix is formed by 3 amino acid per turn of the helix, and it contains 10 atoms including the hydrogen bond. If it is 2.27 ribbon, then the 2.2 amino acids are residue present per turn of the helix, and it is a 7 atoms including hydrogen is present per turn. So this is the way by which you can explain what is the 310 helix or 2.27 ribbon or the pi helix. Now we should go for the second type of structure, that is known as the beta plated sheet. And the beta plated sheet was determined by Pauling and Corey by X-ray crystallography structure. This is the more extended conformation of the polypeptide chain, and is found in a number of fibrous protein called beta keratin. Beta protein, this is the both are similar type of structure of the beta plated sheet. Beta conformation, the backbone of the polypeptide chain is extended into a zigzag. This is the zigzag structure. This is the zigzag structure. Zigzag. You can see that this is the zigzag structure rather than the helical. The silk protein fiber and the zigzag polypeptide chain is arranged side by side to form a structure resembling a series of pleats. This is the pleat structure. That is why it is called beta plated sheet. In beta conformation, the hydrogen bonding can be either intrachain. This is the interchain, or they can be either intrachain. Intrachain means within the chain they can form a hydrogen bond between this NH and CO group, and it can form CO and this NH. So this is the interchain. You can see there, this is the interchain, and they can form strong hydrogen bond between the interchain also. The calculated distance between the two CHR group on the same chain of the polypeptide is approximately this CHR and this CHR. This is about 7.2, but in actual calculation it becomes 7 because of the constraints. Due to crowding caused by the side chain, thereby preventing the side chain fully extended. So the distance between the two CHR groups on the same side is actually 7.2, but in actual measurement it is 7 because of the constraints of the R group. So now you have to understand the fact that what do you mean by the parallel anti-parallel beta plated sheet? In parallel beta plated sheet, in any peptide chain, it is composed of N terminal. One is known as N terminal, and that is known as the C terminal N. So in this particular chain, two chains, it starts with the N terminal residue, so it is a parallel chain. And in this case, it starts with the C terminal N, and this one is started from the N terminal N. That means the C terminal N meets with the N terminal N, and the N terminal N meets with the C terminal N. But in parallel residue, the N terminal N meets with the N terminal N, and the C terminal N meets with the C terminal N. So in this case, the hydrogen bonding occurs by in a slanting position. We can see there this NH and CO groups can form a hydrogen bond, but this is a slanting position. These hydrogen bonds are actually slanted, but here the hydrogen bonds are actually parallel. The slanting increases the distance between the NH and CO group. That is why this parallel beta-plated sheets are much weaker than the anti-parallel beta-plated sheet. That means in parallel beta-plated sheet, the consecutive hydrogen bonds in slanted to each other across the long axis of the strand. In an anti-parallel beta-plated sheet, the consecutive hydrogen bonds are alternatively closer from each other and are approximately parallel to each other and perpendicular to the axis of the backbone. The slanting distortion of hydrogen bond makes parallel beta sheets less stable than the anti-parallel beta-plated sheet. Now the third part, this is the hairpin structure, hairpin connection between the anti-parallel strands. So this is the anti-parallel strands. So they can form within in the plane, or it may be the above the plane, or it can form the below the plane, and this is called the turn structure of protein. And these actually made up of those amino acids, they cannot form alpha helix or beta-plated sheet, or that is alternatively known as the random coil. So random coil is a polymer conformation where the monomer subunits are oriented randomly while still being bonded to adjacent units, and it lacks a well-defined structure. So it means that it is composed of alpha helix or beta-plated sheet. Between the alpha helix and beta-plated sheet, these green colored structures is actually the random coil, which is made up of those amino acids that cannot form either alpha helix or beta-plated sheet. Say a stretch of glutamic acid, a stretch of lysine, proline, glycine. These are the amino acids, they cannot form a good type of alpha helix or beta-plated sheet structure, so they can form the random coil conformation. Hope you have understand the basic three different types of protein secondary structure. Thank you.